Breaking news! New gene identified in the study of multiple sclerosis. There's been a new gene, a nuclear receptor identified as a risk factor for MS. I'm going to take you through it. Paper was published in Neuron just a few days ago. First author is Mr. Wang, and the last author is Dr. Carles Villarino Guel. Okay. First of all, they present a case study of two families, two pedigrees of MS patients, very severe MS patients that runs in the family. So family 1 has got, 6 patients. Family 2 has got only 2. They're both from a Caucasian background. Okay, first of all, figure 1 was the pedigree. Now a bit of details. NR1H3 is the receptor. It's better known as LXRA, liver X receptor A. It's obviously one of those nuclear receptors that has to dimerize with RXR, retinoid X receptor, and from there they will initiate transcription. What they found was only one variant, arginine 415 to glutamine. Yeah, that's right, arginine to glutamine in NR1H3 was not identified in any controls, but was found in the MS families. And segregation analysis confirms that the haplotype of these two families is actually the same. So these two families could actually be descended from one ancestor, in whom the mutation originated. Now they do a bit of genetic linkage analysis. Linkage analysis is always important. So what they are reporting here is the LOD, logarithm of odds, for arginine 415 to glutamine and multiple sclerosis incidence. So they found that their LOD was 2.2. Now I read on Wikipedia that a score of 3 is considered convincing. So take this result with a grain of salt. They try to justify this by saying, well, the pedigrees we studied were small, which is true. There were two very small families. So here's MS family 1, family 2. Black circles are the patients. Some other interesting bits they reported are a GERP score, genetic evolutionary rate profiling score, of 5.13. So they call this a high level of conservation. Certainly from what you can see in their figure 2, here are the sequences of a ton of different liver X receptor A's in different species. Look, C. elegans, they even got C. elegans in here. Zebrafish, that mutation is absolutely conserved. Okay, one other thing they report is a SIFT score of 0. So that's, and a polyphen 2 score of 1. So basically they use this to say that a glutamine substitution is predicted to be deleterious. You know what? I think it's pretty obvious that glutamine from arginine is deleterious. It's a completely different type of amino acid. Actually, at first I thought that said aspartate, and I thought that was bad enough, but arginine is positively charged, right? Guanidine group. Glutamine is not charged, but it's a bit different length too, so steric hindrance. Indeed, they confirmed this. They did some pull downs, and they found that their LXRA could perfectly fine interact with the RXR, but not when it carried the mutation. So here's a Western blot. I'm not sure exactly how it works, but look, pull down, there's their pull down. And does it interact with the receptor? Here's the thing. Okay, cool. All right, figure 3. They report that their mutation, right, glutamine from arginine, is located, these receptors have to dimerize with RXR in order to function. So here's the dimer structure. That's the dimer, okay, top part and bottom part. And you can see that right where the interface is, you've got these two amino acids that project in, and they basically have a handshake. So obviously if you replace that, with that non-synonymous mutation, you're going to completely knock out that interaction, which is required for transcriptional activity. A bit more intuitive way, I guess, of testing that is using a reporter, a transcriptional reporter, I believe. Anyway, a reporter for a promoter. And they use luciferase, right? That's the gene that's usually used for these kinds of reporter assays. And what they found was, okay, plasmid alone, nothing. LXRE, liver X receptor, you get a little bit of an increase with your wild type, and you don't get that with your mutant. And they can kind of sensitize this effect. So here, you get an increase in binding, and then it gets knocked back down with the mutant. And again, here, you get an increase in binding when you administer LXR, knock it back down. This column is with it's with an agonist that will actually bind, I guess, at the interface and make these two proteins bind even more tightly. So that's just a way to sensitize their Okay, that's about all there is in the way of data, but let's take a bit of a look at the discussion. Okay, so what they did find, so this kind of fits in well with some of the other literature that's around about liver X receptors and MS, which actually comes as a complete surprise to me. So look, apparently the LXR-A-B, the liver X receptor A and B double KO mouse, has a brain phenotype. They have axonal atrophy, neuronal loss, astrogliosis, lipid accumulation. Stay tuned for that. That's the next paper. It has to do with lipids in MS. Lipids in Alzheimer's, actually, so not really. Anyway, so what they found was in these double KO mouse for the liver X receptors, they've got, messed up expression of myelin genes, including protein 0, and the myelin sheath by ultrastructure is also it's messed up. Some other stuff they found, that they found from the literature search, LXR and its ligand is an important negative regulator of inflammatory gene and innate immune response. Inter alia, IL-6, INOS, COX-2, some other stuff. So, yeah, basically, I think this study fits in really well. It's well done. There's a bit of even biochemistry in here with the protein structure. Fits in well with the current research in multiple sclerosis. And I think this really should help revitalize the field a little bit. I saw it on Facebook. It's big news. In summary, highlights an arginine to glutamine mutation in NR1H3 causes multiple sclerosis. In families, the clinical phenotype is consistent with progressive multiple sclerosis. The mutation results in loss of function, leading to transcriptional dysregulation, and common variants in NR1H3 are associated with primary progressive MS. Summary. Multiple sclerosis is an inflammatory disease characterized by myelin loss and neuronal dysfunction. Despite the aggregation observed in some families, pathogenic mutations have remained elusive. At least I've never heard of any other than the MHC gene polymorphisms. In this study, we described the identification of NR1H3, arginine 415-glutamine, in 7 MS patients from 2 families presenting severe and progressive disease. Average age of onset of 34 years. Additionally, association analysis of common variants in NR1H3 identified, long name, conferring a 1.35-fold increased risk of developing progressive MS. The P-arginine 415-glutamine position is highly conserved in orthologs and paralogs. Oh yeah, I forgot to mention, basically all the other nuclear receptors have this conserved residue, even the ones with different targets. And this mutation disrupts NR1H3 heterodimerization and transcriptional activation of target genes. Protein expression analysis revealed that mutant NR1H3, liver X receptor A, they just finally tell it even now, alters gene expression profiles, suggesting a disruption in transcriptional regulation as one of the mechanisms underlying MS pathogenesis. Our study indicates that pharmacological activation of LXR alpha, A, or its targets may lead to effective treatments for the highly debilitating and currently untreatable progressive phase of MS. Thanks for tuning in. The second paper I'll present today is entitled Aberrant Lipid Metabolism in the Forebrain Niche Suppresses Adult Neural Stem Cell Proliferation in an Animal Model of Alzheimer's Disease. This paper is authored by Ms. Laura Hamilton and coming from the laboratory of Dr. Carl Fernandez, whom I've had the great honor of just meeting, and he did a very good job of summarizing his results in a presentation. So today I'm going to do my best to try to relay to you the interesting findings within. All right, so given that the title of this paper is about Alzheimer's disease, let me just explain a little bit about the lab's focus. So they use, first of all, they use the triple transgenic mouse model of Alzheimer's disease. So that's, let me think, amyloid precursor protein, tau, and the secretase, presenilin 1. So triple transgenic Alzheimer's disease, 3 human proteins expressed in a mouse, very accurately models Alzheimer's disease. The real focus of the lab is neural stem cells, NSCs, and in particular they study them in 2 locations. One is the subventricular zone, so the layer underlying the ventricles, and second part is the dentate gyrus of the hippocampus. So the hippocampus, memory and stuff. So I guess dentate gyrus dentatus, something like that. What Dr. Fernandez and his team found, figure 1, is they had already remarked that there was lower amounts of proliferation when they looked at these NSCs, neural stem cells, in 2 niches. So I'll present to you the first bit here. So they see lower amounts of Ki67, GFAP positive NSCs, proliferating NSCs, so Ki67 is the proliferation marker. Ki67 positive, GFAP positive NSCs are down in Alzheimer's. Ki67 positive DCX, I think it's double cortin positive, transamplifying neuroblasts, sorry, not transamplifying, but neuroblasts are down in Alzheimer's. Oh, and I guess as a control here, they've got a little bit of just a staining for DCX, so there's no change in that. It's just the proliferation that's changed. Okay, now on to some of the most interesting, exciting stuff in the paper, I think. So, at some point, studying these neural stem cells, they got the idea to stain for, so they got the idea to stain for lipids, right? So because they had heard that, lipids were a big player in the field. So they stained for lipids using this stain called oil red O. And basically what they found was in this, around the ventricles, all these areas around the ventricles, remember the SVZ, the subventricular zone, is a stem cell niche. So in these ventricles, look, wild type mice, these dots that you're looking at, dark dots, not very many, right? In Alzheimer's disease, triple transgenic mice, oil red O positive droplets just all over the ventricle here. And so here you go, lateral ventricle and third ventricle, and they increase with age too. So, that's not so interesting, right? It's just a lipid droplet. Hang on, it's exactly the same in human samples. So they got human tissues, subventricular zone tissues, control humans. Not much there, a few lipid droplets. Alzheimer's disease patients, oil red, oil red, oil red, oil red. Tell me, does that not look exactly the same as the mouse model? Oh yeah, and so they found that these droplets were actually, I'm not sure exactly where it says, but they were within the cells. The ependymal cells are cells that line the ventricle, like endothelial cells, basically. The ependymal cells, healthy mouse and Alzheimer's mouse, only lipid droplets are only found in the ependymal cells. 0 lipid droplets found in the subependymal regions. Guys, it's so exciting. We're only on figure 1. Okay, figure 2. So in this part, okay, so they could have just left it at the oil red, but no, they don't stop at that. They're going to actually go on to use mass spec. They're going to use mass spectrometry on the tissue. So basically, they've got their tissue, right? It's like an XY. It's a 2-dimensional, matrix. It's a thin section of tissue, right? And basically, they're going to do multi-mass spectrometry on every pixel, every small square of this tissue section. And apparently, it's got about 10 micron resolution, so not quite cellular, but okay. Anyway, here's the results. Look, perfectly recapitulates what I showed you earlier. Healthy mice, wild type mice, 3x, triple transgenic, Alzheimer's disease mice. Okay, so here you're seeing the ventricle, right? And all around the ventricular zone in the brain, you've got signal. And each of these columns is basically four different lipids. So they've got, well, four different triglycerides. So they've got 51, 52, 54, 54, So that's the lipid notation. Number of carbons and number of unsaturations. And what does this give, right? What's the defining principle of all these lipids? Are they related in any way? Well, here's basically a little chart of those lipids. Hang on a sec. There's a little chart of these lipids. So basically, there's a bunch of them in a pathway. They get interconverted, but if you notice, there's basically only two enzymes. There's SCD and ELOVL, or basically two major enzymes that are involved in interconverting these triglycerides. All right, figure 3. So basically they found that by injecting oleic acid, so oleic acid was one of the constituents of the triglycerides they found, they could recapitulate both the oil red O positive droplets and this phenomenon by mass spec. So here you go, here's the figure. Vehicle, those are the ventricles, okay? Oleic acid administration. Zoom in on this segment, you've got the ventricle wall here, the subventricular zone. Well, actually the ependymal layer, and there's a ton of oil red granules in here, there's no oil red. Same thing here, you got vehicle oleic acid. You can recapitulate this mass spec. Alright, next they test the effect of this intracranially administered oleic acid on NSC activity. Right, so basically they also recapitulate what I showed you in figure 1. They've got lower numbers of Ki67 positive cells, basically Ki67 EBA1 positive or Ki67 GFAP positive, basically are all decreased in this triple transgenic model. So not only does this oleic acid administration induce the droplets, it also correlates with disease. And there were actually some other really tricky experiments in that figure that I had to skip because of time constraints, but I invite you, please read the paper. Cell stem cell. Okay, so then they wondered whether this oleic acid was going to go through the blood, the CSF, right? Because the ependymal layer is kind of sandwiched between the blood and the CSF. I'd give you a diagram if I had one. Okay, but so they test. Okay, right. So they're going to test the CSF and the blood. So let me show you the results for their, the levels of triglycerides in the blood or the CSF. Are they elevated? No. Here's blood, CSF. They're both, basically not statistically significantly different. They're just, they're about the same. So what do they do? So so maybe, if it's neither of those, maybe it's within the cells. Maybe the cells are, their gene expression is going to be influenced. So I must have isolated, I guess, the subventricular zone. Oh yeah, I remember they microdissected the SVZ, the subventricular zone. Did a microarray. So look at this. Always good to see data as presented as a volcano plot. On this axis is p-value, so statistical probability. On these axes is going to be, fold up or down regulation. So the left branch is going to be upregulated, downregulated. So here's the genes they found. And so basically the answer is, they found quite a bit of a fold change between triple transgenic and wild-type animals. Okay, so basically, to recap, neural stem cells are altered in Alzheimer's disease. Neural stem cells in the subventricular zone and the hippocampus, so memory, right, are slowed down. They're less effective in Alzheimer's disease. This means that there's less self-renewal of neurons, there's less neuroblasts being generated, right? And so these correlate with oleic acid, and triglyceride-based droplets in the ependymal cell layer, oil red O positive, and these droplets, are formed probably by genetic changes within the ependymal, at least within the ependymal cell layer, and not by just increased uptake of oleic acid from the blood or CSF. Final figure 7, I believe. This is the tour de force right here. Basically, back on this page I showed you, SCD is one of the enzymes that converts between the different triglycerides. What do they do here? Treatment with an anti-SCD1 inhibitor not only reverses the effects of all the oleic acid, the different triglycerides levels, but it also restores the capabilities of proliferation to neural stem cells I can't even believe it. So basically, I just showed you a representative bar graph here, but they've got the exact same stuff as in the other figures. Ki67 GFAP, Ki67 DCX, etc. Okay, so the working model. Let me show you the little graphical abstract here. Cell, stem cell, very cute. Blood vessel here, neural stem cell here. These are a bunch of neuroblasts that are being generated from the neural stem cell, which has a capacity for self-renewal. What's going to happen is an increase of SCD1 is going to drive incorporation of these giant lipid droplets into the ependymal cell layer, which is somehow, through an unknown mechanism, going to, well, I think it's an AKT related mechanism, but basically, through an unknown mechanism, this is going to force neural stem cells to shut down. So what do? You treat with an SCD1 inhibitor, lipid droplets go away, neural stem cells are allowed to proliferate, neuroblasts are formed. Memory, hopefully, is restored in Alzheimer's disease patients. To summarize, lipid metabolism is fundamental for brain development and function, but its roles in normal and pathological neural stem cell NSC regulation remain largely unexplored. Here we uncover a fatty acid-mediated mechanism suppressing endogenous NSC activity in Alzheimer's disease, AD. We found that post-mortem AD brains and triple transgenic Alzheimer's disease mice accumulate neutral lipids within ependymal cells, the main support cell of the forebrain NSC niche. Mass spectrometry and microarray analyses identified these lipids as oleic acid-enriched triglycerides that originate from niche-derived, rather than peripheral, blood CSF, lipid metabolism defects. In wild-type mice, locally increasing oleic acid was sufficient to recapitulate the AD-associated ependymal triglyceride phenotype and inhibit NSC proliferation. Oh yeah, so those oleic acid administration experiments I showed you were wild-type. These studies support a pathogenic mechanism whereby AD-induced perturbation of niche fatty acid metabolism suppresses the homeostatic and regenerative functions of NSCs, thus leading to neurodegeneration. All right, thanks for tuning in this week. That's all the time I've got. These two papers, Neuron and stem cell, very big news these days, multiple sclerosis and Alzheimer's disease. So stay tuned. Please tell me if I missed anything, made any egregious errors, and please like and subscribe.